So, SS, we can have the prayer from you. Okay. Thank you so much, Zeo SS, for leading us in the opening prayer. Yes, so allow me to disable. In case there is anything you want to say, I'm going to disable your microphones. I'm going to disable that feature of the microphone, and then I will allow you to be able to do other features. Other features will be left open, but in a special way, allow me to welcome all the schools that are joining in from the different parts of the country. Thank you so much for joining this session. Dear students, thank you so much for joining in, and we want to wish you all the best in this session. We want to thank the Ministry of Education and Sports for supporting this activity of ensuring that we collaborate as schools across the country. We are really very grateful. We want to thank the Research Network of Uganda and all the organizations that are doing all what it takes to see that we can meet as a country on one screen and be taught by one of the schools. So we are really very grateful and thank you so much. In our midst, we have some officials from the Ministry of Education and Sports, and allow me to invite the principal at the secondary department, Mr. Ronald Dungu, who is representing the Ministry of Education, to give us his opening remarks. Mr. Ronald, you're most welcome.
Thank you, Mr. Kaziba. I hope you can hear me.
Yes, Mr. Ronald.
Okay. Good afternoon, students, and good afternoon, teachers, and everyone who is joining in this afternoon. I'm happy that we are meeting once again after a long time, probably as biology learners. We had chemistry sometime last week, but at least as biology, we have not been able to meet lately. So we thank God who has kept us safe up to this period when we present the last candidates in the old curriculum. As you may be aware, we are winding up the old curriculum and we shall now be sticking to the new curriculum. In 2022, we had poor performance in biology, senior 4, and Mama, the Minister of Education and Sports, was very clear that something needs to be done to improve performance. She ordered us to ensure that we work towards improving performance, and I hope the teachers have been working hard and the students are ready. On 7th July 2023, the Association of Biology Educators held an online workshop for teachers, and one of the schools in Arua brought in students. It was very clear to us that we needed to hold online sessions for students. And it is against that background that on 3rd August, we held the first biology seminar for your group with the students and discovered again that we needed to have another seminar to try and close the learning gap. So we are here to close the learning gap. And I want to thank the Forum for Innovative Teachers for organizing this session, and also thank our facilitator, Mr. Arthur Kugonza from the Association of Biology Educators for always offering this support. As Ministry of Education and Sports, we are supportive of e-learning and would like to see more innovative teaching carried on by our teachers. It is now my pleasure to declare this session open, and I'll be here to follow and see how it goes, and then come back at the end as we close. I thank you, Mr. Kaziba, for running this, and please let us now start the engagement. Thank you.
Thank you so much, Mr. Ronald Dungu, for those opening remarks. Allow me now to welcome Teacher Arthur to take us through the presentation. Teacher Arthur, you're most welcome.
Thank you so much, Mr. Kaziba. Thank you so much, Mr. Dungu, for these engagements and for this innovation that has actually opened our eyes that we can share ideas and we can share knowledge without having to meet physically, but you can still do it virtually. So I'm happy to be part of these engagements in our struggle or in our hope, in our quest to improve the performance of biology in the country. My name is Teacher Arthur Kubonza. Once again, I teach of biology. So this is our second practical session. The first time we met, we were going through a paper, and they promised that before we go for exams, we must complete at least that paper to see or to get the skills for what we need to answer practical biology and be able to pass it. We are aware of the challenges in some schools that the resources may not be sufficient to enable the teachers carry out all the necessary practicals that they are supposed to carry out. But I'm hoping that these engagements will at least help bridge that gap, in that by the time we go for biology, that I think will be next week or next week but one, at least we are somehow confident and well equipped what to do, and also to prepare ourselves how to read this biology that for the biology practicals as well. So without wasting time, I'm going to open our paper that we were working on last time, and we start from there. So I hope my screen is coming through. So this is the paper that we are looking at. Of course, we started with our number one now, and we looked at how food tests are done and also how observations are done, and also knowing the idea behind doing some of the things. But today, we are not going to dwell so much on this number one, because I'm hoping that most of the things that we needed, we did them last time we met. But however, I want to highlight more that number one before we go to number two, still using this paper as our guiding paper. Now, number one, it is always about, we call it physiology. Number one, physiology, where you are playing around with the chemicals, you are manipulating the chemicals. That is number one. Or sometimes we call it the chemical number. Now, take note that number one may not necessarily be only about food tests. It can be about other things. So for food tests, at least now we have the basic skills, just knowing the reagent that is used and what that reagent tests for. Also take note that number one, they can also ask you something about catalase enzyme, or they can ask you something about enzymes. And these enzymes, we have catalase, it can be amylase, it can be pepsin, it can be trypsin. So what is important here is to know the substrate and the products of these enzymes here. if in the question you sense that it is about food tests, and in these food tests, you carry out the food test, for instance, the first time, you find when starch are there. So when you find that starch is there, then you see they're telling you to add the chemical. After adding a chemical, then you intubate for about 30 minutes. Now, obviously, you now start predicting that it is likely going to be either amylase enzyme. Why? Because amylase enzyme is the enzyme that catalyzes the breakdown of starch to reducing sugars. Now, in the practicals, starch is broken down to reducing sugars. Though we know theoretically that starch is broken down to maltose. So how do you notice that maybe what I'm dealing with here is amylase enzyme? It is possible. For instance, when you look at this question here, now this question here, just to take you back briefly to know where we started from, those ones who are not able to attend the first session. Now, for instance, when you look at this question here, you carry out food tests. Now, in this solution, starch was present. So means that now here, when you add iodine, you find that starch is present. In other words, here you get a blue-black solution, and you conclude by saying that starch is present. Now, reducing sugars were absent, and the proteins were also absent. Just that is the background to this table A. Starch was present, reducing sugars were absent. Then now they tell students to do this. You pick that solution of A. Mind you, this solution of A, you have, after carrying out these food tests here in the first table, you have discovered that it is only containing starch. So you add another solution B whose identity has been hidden. And when you add this B to A, then they say add sodium hydroxide. Then you repeat the same procedure, you add hydrochloric acid. Now here you start suspecting. Of course, you know the identity of solution A, that is starch. So they're telling you to add this at solution B, then you add sodium hydroxide, in another one you add hydrochloric acid. Then they tell you to intubate for 30 minutes. Now, intubating means you get these things in a test tube. Let me just pick something here. Okay. Intubating means, I hope my video clip can go through. You prepare your water bath maintained at 40 degrees Celsius. So you get maybe your beaker. In this beaker, you put there the water, and you use a thermometer to measure the temperature of that water. Now, when you realize that your temperature is at 40, when you realize that your temperature is at 40, then it means that now your water bath is ready. Then you bring your contents in the test tube. And in this case, which contents do we have? We are having 2 cubic centimeters of solution A, 1 cubic centimeter of solution B, and 1 cubic centimeter of sodium hydroxide. So you dip your contents in that beaker. Then you leave that setup to stand for 30 minutes. According to the question, some questions can say 20 minutes, other questions can say 25 minutes. Then you proceed with answering other questions. So that is what they call intubation. That is to intubate. Now you keep on monitoring your liquid, the temperature of the water. If it drops below 37, you remove the test tube contents, the test with its contents. You add there little hot water, you stir. After that, you see if the temperature has gone back to 40, then you replace your test tube with the contents. Now, that is what they call intubation. So they're saying that now, when you do that, then you continue following instructions, reading instructions. So here they are saying that 1 cubic centimeter of the contents, sorry, of the contents in the test tube, you add 3 drops of iodine solution. So it means that now in your solution, this solution here, you get the test tube 1, you divide it into 2. After dividing it into 2, in 1, you don't, in this case, you don't divide it into 2. Sorry, the instructions here say otherwise. So in this first test tube here, after intubating for 30 minutes, now you add iodine. That is what the instructions are saying. Add 3 drops of iodine. What do you observe and what is the deduction? Now, to cut the long story short, you may be surprised if you find that in A, starch will be absent now in A. So here you're likely to get starch absent. Then in test tube 2, you're going to get starch present. So it means here starch was lost. Remember, at first it was there. Then here starch is still present. So meaning that now something happened in test tube 1 that resulted into the loss of starch. Then in test tube 2, starch was never worked on, so it remained there. Now you start probing what could have happened in this case. So then that's when you start suspecting that maybe there was something with this solution B. Solution B could be the one responsible for the starch that is lost. Now that's when you start to think that now you are dealing with the enzymes. You are dealing with enzymes because enzymes are the ones that are responsible for loss of food nutrients. Then now you start asking yourself now, which enzyme works on starch? Then that is amylase enzyme. Then now you suspect, or you know that now, whatever you're working with, solution B must be an enzyme that works on starch. Now, what of these ones here? Sodium hydroxide and hyaluronic acid. Now, in enzyme experiments, when you use sodium hydroxide and hydrochloric acid, this is now introducing the effect of pH into your work, effect of pH. So it means that either, because we know enzymes work under specific pH, they either work in acidic pH or they either work in alkaline pH. Full stop. So it means that now sodium hydroxide is introducing alkaline pH. Hydrochloric acid is introducing acidic pH. So if that one has happened, then you'll know that it means this enzyme here in B works best in alkaline conditions, but does not work in acidic conditions. Why am I able to make that conclusion? Because I have seen that it is in test tube 1 where starch was worked on, where starch was lost. So those are the indicators. Then now you now start answering these explanation questions. The mistake that you candidates normally do, you find instructions like these ones. Then what you do, you just read and just follow instructions without knowing what actually you're doing. You read here, they are saying to this solution, add this, you go and add. Whatever they tell you, you go and do. Here they tell you, okay, add 2 cubic centimeters of A, add 1 cubic centimeter of B, add 1 cubic centimeter of sodium hydroxide. You also go and add. They have told me add this, add this. Then what next? They are saying shake the contents well, intubate, you intubate. After 30 minutes, remove, you remove. Then you do this. Then when you come here now, you ask yourself now, what am I doing? You don't actually know what you are doing. What you always ought to do when you find a question like this, first read through the whole question without touching anything. Read through the whole question. Try to pick out the concept. Which concept am I looking for? Or which concept am I testing for in this number? After getting the concept, then now you have your own predictions that, oh, now this one, the table 1 is about food test, okay? Then table 2 Or part B is now about catalysis or intubation or maybe enzyme 1. Then there now you start now still developing the theory. What is this? When they say add sodium hydroxide, you ask now, why should I add sodium hydroxide? Or what is sodium hydroxide introducing into this experiment here? Oh, so it means it is about maybe the pH. Then what of hydrochloric acid? This one, it means, oh, it is introducing acidic pH. This one introducing an alkaline pH. Then, okay, now, when I intubate, what do I expect to get? You first ask yourself, what do I expect to get before you carry out the video experiment? Then after knowing actually what you're supposed to do, and you also have an idea what you expect to get, and then now you go and now carry out or follow the procedures. When you follow the procedures, now you'll be knowing what to do. You'll be knowing what you're doing. When you add hydrochloric acid, you'll be knowing that, oh, I'm now adding this hydrochloric acid because it is introducing acidic pH. I'm now adding sodium hydroxide because it is introducing acidic alkaline pH. Like that. Don't just read instructions wholesomely, as if you're just reading, for instance, maybe as if you're just reading something, or maybe you're just reading a story. No, know what you are doing, or know what you're expected to do, then go ahead and do it. Otherwise, if you just follow instructions, you'll get lost along the way. So I will just leave it at that. But also in these number 1 questions, there are other enzymes, that is pepsin and trypsin. So now, pepsin and trypsin, when do you know that you are now actually working with pepsin or you're working with trypsin enzymes? They tell you to carry out food tests. And in these food tests, you discover proteins are present. Then when proteins are present, then they go ahead and they say, okay, part B, intubate. After intubating, so the part B, add maybe solution Q or solution X, then intubate. Then after 30 minutes, test for the proteins. Then you find out of the blue, the proteins have now disappeared. Yet at first, they were there. Then now you start suspecting that now, whatever I'm dealing with here is an enzyme that works on proteins. And these enzymes are the trypsin and the pepsin. So that's where you know, that's when you know that you're working with pepsin or trypsin. Then another interesting enzyme is this catalase enzyme. Now, catalase enzyme, this one, you don't even have to ask yourself, when do I know? The moment you enter, maybe the moment you see that I've provided you with the hydrogen peroxide, Irish potato, liver, those things, or maybe raw pawpaw, then you know you're now working with catalase enzyme. That's when you get to know that, oh, they're now dealing with catalase enzyme. Now, to talk briefly about this catalase enzyme, what is this catalase enzyme and what does it do? Now, for catalase enzyme, let me open the notes. Let me open the notes and we see. Yeah, those things. Okay. Yeah. I hope my screen is coming through. These are just notes. You see everything, these practicals, everything is taught in theory. And then when they extract out those theoretical parts, then they bring them in practicals. So I want to scroll where there are enzymes. and specifically catalase enzyme. Catalase enzyme is here. Now, of course, I'll just also talk about it briefly. Now, catalase, like we said, catalase enzyme, for it is unique. Catalase enzyme breaks down hydrogen peroxide. And hydrogen peroxide is the chemical that is always provided in case it is a practical and they're asking about catalase enzyme. Now, and this catalase enzyme cannot be given to you as a solution. You extract it or you use tissues that have this catalase enzyme. And these are the tissues: liver, meat, and Irish potatoes. So they give you maybe Irish potato, or they give you a piece of liver. Then in this piece of liver, they maybe give you instructions: cut the Irish potato into small cubes, then drop those cubes in a solution of hydrogen peroxide. Then you see bubbles forming. You see bubbles forming. I used to have those, let me open this. Let me open those PowerPoint things. Then we see the bubbles that I'm talking about, because I know some of you have never done an experiment about this hydrogen peroxide. It is here. The catalase enzyme. Let me also use these PowerPoint slides to help me go through that catalase enzyme. So we say that it breaks down hydrogen peroxide into water and oxygen gas. And that enzyme is found in Irish potatoes. So what happens? Now, when you add anything like maybe liver or maybe Irish potatoes into hydrogen peroxide, this is what you see. You see bubbles forming, or you see a froth being formed. Now, this is when you know that actually you're dealing with catalase enzyme. Then now, there is a known format of how we record observations involving this catalase enzyme, and also how we conclude. There is that known format. Now, this format is like this. I'll take you back to the notes. You see, when you look at this table here, so they can say to one test tube, to test tube one, of course, in this test tube, we already have hydrogen peroxide. Then they say, add one whole cube. So this cube is the cube that you have cut from the Irish potato. Then when they say, add this one whole cube, you see what happens. Now here we are saying that there is a slow bubbling. Or you can even say few bubbles formed. Then where you add one cube maybe after cutting it down into 16 equal parts, it means that now you have got that, you've got your Irish potato, 1 cube, then that cube you cut it into tiny parts. Then you see rapid bubbling. But now, how do you see this bubbling, or how do you phrase them? You see, we're using the word slow, and here we're also using the word rapid. Now, this is what you do. Now, when you look at the pictures on the screen, when you're carrying out experiments of catalase enzyme or hydrogen peroxide, the first thing to do, make sure that you first prepare all the cubes that you need before dropping them into the test tubes. You prepare all the cubes that you need before dropping them into the hydrogen peroxide solution. Then after preparing all those cubes that you need, then you get your test tubes. If maybe they said add hydrogen peroxide in 5 test tubes, you do it. Then after that, they say now drop. Now you after measuring your hydrogen peroxide into the test tubes, then you get now your prepared cubes. You drop them in their respective test tubes almost at the same time. You do it almost at the same time. If here they wanted, if in test tube 1, for instance, in this test tube, if in test tube 1, they wanted you to put there 1 whole cube, you put it there. In test tube 2, maybe they want 1 whole cube, but after cutting it into 4 pieces. Now, by that time, you'll have already cut them into 4 pieces. Then you carry them, you put them there, or concurrently, or at the same time. Then after that, you sit down and observe. You sit and observe. You see now, how is the hydrogen peroxide reacting with the cubes that I have dropped into the test tubes? You ask yourself that. Now, then after that, now you're going now to write observations and also make conclusions and conclude. Now, in this one here, when you look at the way I arranged my test tubes in this picture here, you see they are following a trend. When you see here, in my first test tube, you see the level of froth is small. Then in the second test tube, you see it is slightly above the first one. Then in the third, you see it is above the second one. The fourth and the fifth, like that. You can see even on the fifth, the froth is about even to pour. The froth is about to pour. Now, if the froth is about to pour, then now the words to use, you have to develop words to use which are comparative. Because the words we use are fixed. At the end of the day, we must say that maybe there is a moderate bubbling, rapid bubbling, or slow bubbling. At least that is what you can see is happening with the way I wrote my observations in the notes. Slow bubbling, rapid bubbling. But now, if I look at this, how do I tell that this is rapid? How do I tell that this is slow? Now, I will arrange my test tubes like this. Then I will now pick words to use. How will I pick them? I'm going to use annotations. Let me write there. Now, I will say, for instance, in this first one, if I say this is very slow, this is very slow. So I'll say in my first one, I'll use the word very slow. Then in the second one, which word follows very slow? Slow. Like that. Then in the third one, I will now use the word from slow, I will say rapid bubbling. We shall also see later other terms to use. If here I say rapid bubbling, then this one must now be very rapid. Very rapid. Like that. Now, what will I use for this one? So I have to go back and rearrange this one. So here, instead of saying from slow, I will go now to moderate. From slow, I'll go to moderate. Then from moderate, now I'll go here to rapid. Then from rapid, I'll now go to very rapid. Like that. So it means when I'm describing or when I'm writing the observations for, say, maybe this middle test tube. So I will say there was moderate bubbling, like that. Or moderate bubbles formed. That is when I'm writing observation for my third test tube. Now, when I'm writing my observation for this last test tube, where the froth is almost pouring, then I will say there was very rapid bubbling, or very many bubbles formed, like that. So that is how you choose the words to use. If there are only 3 test tubes, then you can decide even to start from slow, because there are 4. If there may be 3 test tubes, then you may now choose to use the word slow, then rapid, and very rapid, or maybe slow, moderate, and rapid. So that's how you select the choice of words to use there. If it is a hydrogen peroxide. Then let me now still go back to my presentation. Let me first clear this. So I hope now we have known how we select the choice of words to use to describe or to record the observations to these to the things we get when we're dealing with the hydrogen peroxide. Then now look at this one here. Let me give you more choices of things to use. Okay, let me first switch now to this presentation of mine here. So look at what I was saying here. And first reduce this one here, that every word is visible. So I'm going to use these presentations concurrently. Okay, I think everything will now be visible here. Okay. So you see what I was talking about here. You can either use the word few number of bubbles, or slow bubbling, or slow effervescence. You can choose to use the word very many bubbles, or very rapidly bubbling, or effervescence, like that. So that is how you record those observations for enzymes, sorry, for experiments involving hydrogen peroxide. Then now, how do you conclude the deduction? Now, in the deduction, of course, here there are very many words used here, but if here you said slow bubbling, then in the deduction you can say slow breakdown of hydrogen peroxide. Just that. You don't write there very many words. Slow breakdown of hydrogen peroxide. If you use the word rapid, then it will be rapid breakdown of hydrogen peroxide. Where maybe there was no bubbling at all, you didn't see any bubbles, you will say no breakdown of hydrogen peroxide. Now, if maybe you saw moderate bubbling, then now the deduction would be moderate breakdown of hydrogen peroxide. In other words, the quantification word you use in the observation is the same word that you use in the deduction. If you used very many or very rapid, then in the deduction it will be very rapid breakdown. If you use the word rapid or maybe very slow, then the deduction would be very slow breakdown of hydrogen peroxide. So that is how we deal with the numbers or with the observations and deductions related to hydrogen peroxide, like that. Then also take note that still on this hydrogen peroxide, they can also ask you to give the effect of temperature, the effect of pH. Now, if it is about temperature, they can maybe tell you to boil one of the cubes. So one of the cubes, before using it, you boil it. Like you look through this test here. Boil one whole tube for 10 minutes, drop it in cold water, and after, place it in test tube 3. So if you do that one, then you know that the boiling is going now to kill or to denature catalase enzyme, because it is the Irish potato cube or it is the liver cube that you're using that is containing the enzyme. So if you boil it, then you have denatured that enzyme. Therefore, you don't expect to get bubbling, or you don't expect to get any bubbles. That is what happens. Then pH, they can still tell you that maybe you add a cube. After adding a cube, you add hydrogen peroxide, then you add sodium hydroxide, or you add hydrochloric acid. So if you do that one, then obviously you know you are investigating the effect of pH on catalase enzyme. Then the way you explain be depending on whether you got moderate bubbles or whether you got very many bubbles, depending on that. If you added sodium hydroxide to hydrogen peroxide, and then you added the tube, and there was moderate or there was little or slow bubbling, then when you added hydrochloric acid in a similar setup, you got rapid bubbling. Then you can be able to conclude and say, it means that this catalase enzyme works best in acidic medium. Why? Because when I added hydrochloric acid, there was rapid bubbling. But when I added sodium hydroxide, there was slow bubbling or slow effervescence. That is how you explain those things. So that's how you interpret them. Then that is it now with the enzymes. So still in the notes, you'll go and read more about these catalase enzymes, the do's and the don'ts when you're doing these catalase enzymes, what to take note of, Okay. Then another thing they can ask on that number one is osmosis or diffusion. Osmosis, they can give you viscing tubings. At least I remember last time we saw a viscing tubing, and even we used that viscing tubing, I remember very well. We saw that viscing tubing, investigating the process of osmosis. So these are the main areas where number one can come from. If it is not about food tests, it is about enzymes. It is not about enzymes, then it is about cell physiology, that is osmosis and diffusion. So you be prepared for any of the three, prepared for anyone from these three categories. Come next week when you're sitting, come next week, but one, when you'll be sitting for your exams. But also, some years, there are even some years when they bring soil practicals. But nowadays, these soil practicals are more appearing in agriculture than in biology. But also in biology, if you find practicals about soil, I've given you soil, they want maybe to investigate, they want you to investigate things of capillarity, they want you to investigate things of drainage, the experiments that are carried out or that we have in the notes of soil, they can bring those experiments in practicals. If you find it there, still don't be surprised. Be able to use the theory knowledge to be able to answer those practicals. So that is all with number one. So briefly, that is what I want to talk about, that number one, because it is a very interesting number, though it was not what I had prepared for today. That is why even we have not been able to do any demonstration from these ones. Last time when I was prepared for number one, we did many demonstrations and you're able to see the observations with me in the lab. We're remaining with 6 minutes to 4. We shall finish at around 5:30. Now, I want to use this remaining 6 minutes about physiology. If you have any question about physiology, please, you can ask it now. Any question about physiology in the next 6 minutes before we now switch to another section. So is there any question or is there anything you wish to know about physiology numbers, about number one? If you have any question, you can go to the reactions, then you click on the symbol with the hand up, we shall be able to get it. Or maybe you can even tentatively write it in the inbox. So let them flow in the inbox. Or still you can ask to be unmuted and you air out your question. So there is some questions, some highlights on physiology, if germination. Yes, that is another area which I had somehow ignored. Now, still in that team, number one, they can test you about germination, about physiology. But how about what they exactly test for in this physiology? It is still related to food tests. Food tests. They can give you extracts. They say the solutions given are food extracts, or maybe are extracts from seeds that have germinated for a specified number of days. So they give you a seed, maybe an extract, that this extract has been got from a seed that had been germinated for 3 days. This extract has been gotten from a seed that has been germinated for 1 day. This extract has been got from a seed that has been germinated for 60 days. Then they ask you to carry out food tests. They say maybe carry out the iodine test, carry out the reducing sugar test, carry out the proteins test. You give observations and you give conclusions. Then they now ask you to briefly explain your observations. Now they have to go back and scratch your head. You now go back to the notes. You go back to the notes of growth and development. Of course, in exams, you don't have the luxury of having the notes or maybe the textbooks to go and check. But I'm just saying, when I say you go for the notes, you have read the things. You have read the things in the content. In the head, you have them. So you go, it means you go under the process of germination. What happens during germination that is responsible for the changes in the food tests? So for the changes in the food nutrients in these seeds. We know that seeds store food. And in most cases, they store this food in form of starch. So if they store food in form of starch, why do they store that food? They store that food so that they can use that food when they are germinating. Because remember, seeds don't have leaves to make their own food. So before they make those leaves, they need some food. So they depend on that food that they have stored in the seeds or in the cotyledons, if it is the maize, in the endosperm. But how do they get this food? They get the starch. They have the enzymes, by the way. They get the starch. They hydrolyze that starch to producing sugars or maybe maltose or glucose. Then this glucose is now transported to the embryo of the same seed. And this embryo is used for, this embryo uses this glucose for respiration. The respiration generates the energy, and this energy enables now the embryo to form or to grow. Let me just keep it short, to grow. So it means that now, how do they access the food? They get the starch, convert it into sugars, into glucose. Then this glucose is used in respiration. So it means when you crush a seed which has not yet been germinated, you'll find a lot of starch there. Then when you crush a seed that has already germinated, you don't expect to find the starch there. Why? Because the starch had already been converted to glucose, and this glucose is now being respired to provide energy for the seed to germinate. So that is the indication. So it means that now the amount of starch will go on decreasing as the amount of reducing sugars will go on increasing. Why? Because the stored starch is converted to glucose or reducing sugars. And this glucose is respired to provide energy for developing or for the growing embryo. So that is the science behind giving you extracts from seeds that have germinated for specific days. So if you find that maybe in extract A, starch was not there. Extract B, starch is there. But they're telling you that these are seeds that have been germinating. Now, that is the idea that you use. I hope we have somehow got it. So the seed, for example, we have a seed that has germinated for 1 day, another seed 4 days, another seed maybe 10 days. Now, what do you expect to get? In the seed that has germinated for 1 day, you don't expect to find there reducing sugars. They may not be there. But you'll find there a lot of starch. In the seed that has germinated for 4 days, you'll find there a lot of reducing sugars now, a lot of reducing sugars, and you will find little or no starch in that seed. Why? Because in those 4 days, the starch has been converted to glucose. Then in the seed that has germinated for maybe 10 days, there you will not find there any starch. You will not find it there. The seed, it will not be there. Why? Because all the starch has been converted to glucose, and the glucose has been used for to provide energy. So for respiration, to provide energy for the seed to germinate. So that is what is all about germination physiology. Still in number 1, it is incorporated into the food tests. Then, so I hope I have responded to that question well. Then there is also another question that is there a possibility of combining all let me highlight this. Let me make the chart big. Okay. Okay. This one is saying, is there a possibility of combining all the three in a paper? I don't know what you mean by combining all the three in. Okay, I get it. Now, you're saying the probability of combining enzymes, food tests, and physiology, and cell physiology. It is very possible. They can bring a number. When this number, you're going to carry out food tests. Then after carrying out food tests, it is going to involve an enzyme. And after involving an enzyme, then it's going to involve diffusion. Those numbers are there, diffusion. So how can they be sketched or phrased? They give you a solution, they give you a visking tubing. Then they say, in this solution, add this. Maybe it is a starch. In the starch, you add the solution. And when this solution is maybe, say, amylase enzyme, then this amylase enzyme will now break down the starch inside the visking tubing into reducing sugars. Then reducing sugars will now diffuse out of the visking tubing. It is there to combine all of them. And our first session, I think that is what we talked of when you're demonstrating using the visking tubing. Then someone is asking, what is going to be reducing sugar, protein, and starch content in 1 day, 4 days, and 7 days old seedlings? Still, the other thing about the germination. Now, first of all, if it is practical, you don't have to worry about what is the quantity. Of course, you're carrying out these practicals yourself. They give you an extract from seeds that have germinated for 1 day, and they're asking you to carry out food tests, reducing sugar tests, proteins tests, and starch tests. For you, follow instructions and do it and see, do I have these nutrients? If they are there, then how do I explain these observations that I have made? And the observations I've told, the explanations I've told you, you just have to be knowing the theory behind these practicals, these experiments. If after 7 days, you have found when the proteins are not there, then what does it show? It means that all the proteins were broken down and used. Mr. Kaziba?
Yes, teacher.
Please, you remove that person called George with funny spelling.
Oh.
You remove him from the sessions. Okay. So that's it. So you know that if proteins were present in 1 day, they are present. Then after 7 days, they are absent. Then what happened to them? They were broken down or they were hydrolyzed and used to make new plant parts like that. Reducing sugars. Do you expect them to find them after 1 day? No. Why? Because the hydrolysis of starch has not yet started. After 7 days, then you expect to, after 4 days, you expect to find them there in plenty. Why? Because most of the starch has now been hydrolyzed to reducing to reducing sugars. That's where they are present. Then Mazolido College, they're asking, what can happen to the visking tube when the concentration of solutions is higher than that of the water? Now, still, if it is practical, you see what you observe, you do it and you see. Now, the reason why we give you these practicals is, are you able to follow instructions and report to us the observations? For you, if they say, put a concentrated solution in the visking tubing, Mr. Kaziba? Mr. Kaziba, could you be there? it seems the link was accessed by people. Let me first remove this one.
Teacher Arthur?
Now, Mr. Kaziba, what you're going to do, Mr. Kaziba.
Yes, Teacher Arthur.
Now, kindly you stop, you change the settings there,
Yes,
and
Mr.
stop sharing of videos.
Okay.
You stop sharing of videos.
And also, whoever is not showing the school, we shall remove you because we don't know who is here.
I have.
If you're failing to show to rename, send it in what's in the chat, and we'll help you rename.
All those ones that are using funny words, they are disorganizing. Then I think we can now disable all the videos. Then I will only allow questions from participants whose name is reflected as a school. Okay.
I removed that one. Don't worry.
Okay. Then let me go back now to the questions. Then in the questions, where was I? So I was still on what can happen to the visiting tubing when the concentration of the solutions is higher than that of the water. So for you still, when you're carrying out the experiment, you be very observant. They've said, get the solution, put it in the visiting tubing, and put it in water. Now, using the principles of osmosis, that principles is, that osmosis is the movement of water molecules from a solution, from a dilute solution to a concentrated solution, like that. So what do you expect to happen to the visiting tubing? The visiting tubing will either increase in size or will decrease in size. Those two, depending on the direction of osmosis. Then from there, you'll be able to explain why. The explanation will now be based on because water molecules moved by osmosis from a solution of from a dilute solution to a concentrated solution. Then the central school, where they are asking, how does catalase concentration vary in germinating seeds? Okay. Now it's like now we are going to go into now the theory. Now, for the interest of time, I will just respond those questions that are mainly more into the practical part of it that can be examined practically. But however, if the final something are related to how does catalase concentration vary in germinating seeds, then it is about metabolism tissues that have high metabolism. You expect to find there high amounts of catalase. Then tissues with the low metabolism, you expect to find there little catalase enzyme. And then what happens when a plasmolysed potato cell is placed in distilled water? And now still that one is theoretical. So yeah, still you look at the concept of osmosis, knowing that osmosis, water molecules always moves from a dilute solution to a concentrated solution. And if it moves, if water moves into something, it increases in size. Then in how many ideas can catalase action be investigated? And what are the most appropriate observations and deductions for each idea? That is a very interesting question that from Central School Hoima. Now, in how many ideas can catalase enzyme be investigated? They can give you Irish potatoes. They can give you liver. Then they now ask you to add those tissues to the hydrogen peroxide. Then they ask you now to compare which one has high rates of metabolism, or in which of the two gives a lot of bubbling, like that. Then it should be able to tell that maybe liver tissues have a lot of bubbling, meaning that now they contain a lot of catalase enzyme, because animals are more metabolically active than plants. Then they can still use litmus paper. Litmus paper. They ask you to add those things, following instructions. Then you add litmus paper. If the blue litmus paper turns red, then you know that now the conditions there were acidic. Then now the question would be, then still, what was the extent of the bubbling in that experiment? If the bubbling was very rapid, then you know that now when I'm giving explanations, I have to use the aspect of pH, that there was rapid bubbling because the conditions were acidic. Therefore, this catalase enzyme works best in acidic medium, like that. If maybe you added the red litmus paper and it turned blue, like that, so it can be still asked in that way, using litmus papers. Or we may not use litmus papers. We may add hydrochloric acid and may also add sodium hydroxide. The moment you see that we are having sodium hydroxide and hydrochloric acid in things to deal with the enzymes, know that it is about to temperature. Then they can also tell you to measure the levels of froth, so to measure the size of the froth that is formed. Like when you look at this one here, they can say this one, let me first put back this one. Now, they can give you this, of course, following instructions. And you find that those instructions are directing you towards measuring the level or the size of froth, of the froth formed. Only the level of the froth. So it means you get a ruler, you measure the distance from here up to here. How many centimeters are those? You record. From here up to here, how many centimeters? Sorry, I thought I was sharing the screen. Let me share the screen. Okay, so I hope my screen is coming through. So I was saying that they can ask you to measure the level of the froth. Now, like for example, you've got this one, you find that instructions are asking you also to measure the level of the froth. So you get your ruler, you measure from here up to here. What is that? How many centimeters are those? You record. Maybe from here up to here, how many centimeters are those? You record. Then from that, they ask now to give conclusions. So conclusions would be where you see there are a lot of froth, where you see maybe the level of froth is big in size, that means that now that one, the rate of reaction was very rapid or rapid. Then you're able also to make conclusions based on that, like here. So height may be maximum to brim of the test tube. Then, like for example, now when do we write maximum to brim of the test tube? When you look at this, realize that now, for example, if in my last test tube, the froth poured, like the way you see here, this the middle picture, the froth poured. Also, maybe in this test tube, the froth poured. Yet they want you to measure the size of the froth. So in that case, you cannot measure. In that case, what you just say maximum to brim of the test tube, like that. But why it did not pour, you give us the real measurements. Of course, where there was no bubbling or where there was no froth formed, even if you see there are no bubbles. Now, what is the size of the froth? You just put there the size of the froth. The froth is zero because there is no froth. Only that, don't say that no froth. Just say 0. Then, so those are the ways how the experiments involving hydrogen peroxide or the concepts can be investigated. Then some highlights on osmosis using Irish potato, recording the observations, aspect of volume, length, and weight, and maybe also using non-wood plants. Now, in O level, there are some concepts that they cannot ask you. Like non-wood what? Maybe let me open the notes and we see. Now, what they maybe I think what is asking about related to this. Okay, I hope my screen is coming through. Let me go to osmosis. Now, osmosis and the Irish potatoes. Now, osmosis, it can be asked. By the way, these practicals, they come from the theories that we do in they come from the experiment. So, there are these experiments here. This one here. Experiment demonstrates Tagger and plasmolysis in plant tissues, like this one here. Now, the Irish potato and osmosis, if they example, if they give you a cork bone, you are going to record your observations in a table related to this one here. I also remember having those PowerPoint slides. If us go there briefly. Something like, not that one. Something like this. So you do your instructions, and when you follow your instructions, you find that they are pointing towards this. You get your cork powder. This is a cork powder here. You punch it into the Irish potato. Then you create cylinders. And you find that maybe the instructions are asking you to measure these cylinders here. You measure the length. Then after, they are now asking you to place them in solutions like that. Then they now ask you to make observations like this. Maybe you after maybe waiting for length, then you measure the change in the change in length. Then you convert it to percentage. Then, so obviously, how do you explain those observations? For instance, if maybe, for example, let us assume that this cylinder here maybe it increased in size. Just assuming if it increased in size, let us assume it is maybe this one, the one put in pure water. Now, if it is put in pure water, you can see it increased in size. Now it will be up to you to ask yourself, why does it increase in size? So automatically, here you are investigating things related to osmosis. And the only way such a cylinder can increase in size, it is after absorbing water by osmosis. So it absorbs water by osmosis. Why? Because the cell sap inside it is less concentrated than the pure water where you have put it. Then when it absorbs water by osmosis, obviously it increases in size. Then after increasing in size, that's why you observe this percentage change in length. So that is how you explain those things. For example, you can see, for example, they say here the cylinder in water increased in length because water molecules moved into it from the surrounding water by osmosis, because the cell sap had a higher concentration than the surrounding water. That's how you go about those practicals. But bearing in mind that in practicals, when they ask you to explain concepts, these are the things we are looking out for. The observation, state what you saw. In this case, what we saw was the cylinder increased in size. Then after stating what you saw, are you now able to tell us how it happened? If you're saying that the cylinder increased in length, how did it increase in length? In osmosis. Then if you also tell us how it happened, then are you able now to tell us why it happened? Why did it absorb water by osmosis? Then you now give the because the cell sap had a higher concentration than the surrounding water. So in this physiology, for everything that you explain, in your answer, we are looking for three things. Tell us what happened, how did it happen, and why did it happen? Those are the things that we are always looking out for in numbers, in the explanations of in the explanations involving explaining observations of physiology. Then another question. Let me see here. How to measure 1 centimeter of solution in a test tube? That is a very interesting question. How do you measure 1 cubic centimeter in a test tube? Now when they say that measure 1 cubic centimeter of solution in a test tube, you don't have to get a measuring cylinder. You don't have to get a ruler, for instance. No, you just estimate using your eyes. For instance, let me first get a test tube here. Then I show you how things can be done. Okay, I have got one here. So I'm assuming this is now my test tube. Now, 1 cubic centimeter for you, you just add something little. And remember last time I told that when you're adding these things, when you're mixing these chemicals or adding anything into the test tube, make sure that you're doing something in front of you and at eye level. Your eyes must be able to see the mouth of the test tube and the bottom of the test tube to see what you're adding there and how you are adding it. So eye level, that's where you keep those things. So I'm going to just add something something little like this. This is worth 1 cubic centimeter. You don't have to get, unless if maybe it is about physiology, like osmosis, and it is involving in this cubic. If they say measure 3 cubic centimeters, then you must get a measuring cylinder and measure the 3 cubic centimeters. But for food tests, estimate just something little. If you use a lot of reagents or a lot of solutions, you like something little, you just estimate. Then another question is saying, okay, so I think there are no more questions. That is great. So let us now go to our let us now go to proceed with our paper. So let me share my screen. What is the paper? The paper you're looking at is here. Okay. So in this paper, they are saying you are provided with the specimens R and S, which are flowers. I know students normally fear practicals involving flowers. So they are saying you're provided with specimens R and S, which are flowers. Examine the specimens and give reasons why they are flowers. Now, in practicals, whenever they give you a specimen and they want you to give reasons why it is a flower or why it is what it is. Of course, now this is my flower here. This is my flower. Let me highlight this one. So this is my flower. Now they're asking me, what is this? Of course, you can't call this one a leaf. It is a flower. Then now they're asking you, give reasons. Why are you calling it a flower? So I look at those features it is having. I look at the features it is having, the basic features. Like it is a flower because when you look at these ones, everyone knows that these ones are petals. Is there any organ on a plant that has petals that no? So it means that now I'm able to, this is a flower because it has anthers. This is a flower because it has calyx. Calyx, this the green part. That green part there is what they call the calyx. That green part is what they call the calyx. So those are the reasons why we think this is a flower. And so when they ask you to give reasons why you're calling something so, you just give the distinguishing features it is having. It's what you give. So it means that now, in this case here, so it means that now in this case, to this question here, if they say, examine the specimens and give 4 reasons why they are flowers. So I'm just going to look at the specimen. I will say, first of all, it has petals. That is one reason. It has sepals. That is another reason. It has stigma. That is another reason. It has anthers. Like that. That's what we give. When they ask you for the identity of a specimen, and then they ask you to give reasons for that identity. For instance, they can give you an insect, and they say, maybe they have given you a honeybee. They say, identify the specimen. You say it is an insect. Then they say, give reasons. Now, why is an insect an insect? It is an insect because it has 3 pairs of legs. It has 3 main body divisions. It has 3 thoracic segments. That is why it is called an insect. So that is what they always ask for. When they give you a specimen and they ask you, identify it and give reasons for the identity. Just pick out the basic features that this specimen has. If they give you a leaf, for instance, they say, identify the plant organ, and you say it is a leaf. Then they ask, give reasons for your answer. Then you look at now the features of the leaf. It has green lamina. It has veins. It has a margin. That's why you think it is a leaf. So those are the things that they are always interested in. Then now for this one, they're asking, give a reason in each case, state the type of pollination of each specimen. Now, pollination, I'm assuming, suppose I have these 2 flowers. So you're going to see, to identify the type of pollination and the reason why we're thinking it is that. So let me again first highlight my video. Okay, so I have 2 flowers here. I'm going to assume it is my specimen S and my specimen R. I'll start with this one. Now, this flower here, this flower that you're seeing here, first of all, if they ask you the mode of pollination, definitely I will say that mode, it is pollinated by insects. Why do I think it is pollinated by insects? Because it is brightly colored. That's the reason. But the mode of pollination is different from the type of pollination. We have 2 types of pollinations. We have self-pollination and cross-pollination. So here it's like now they're asking, is this cross-pollination, so cross-pollinated or self-pollinated? Now, the trick is in knowing the definition of what pollination is, and also the position of the anthers and the stigma on a flower, like that. So we know that pollination is the movement of pollen grains from anthers to stigma. So pollen grains are moving from anthers to stigma. So if you look at a flower like this one here, these ones on top, I hope my video is highlighted for everyone to see. Okay, it is highlighted. Now, if you look at this flower, this part is the stigma. So this one actually has 5 stigma lobes, these ones here. And when you look, then these ones, the ones you see appearing yellowish in color, these are now the pollen grains. So it means that now they are on the anthers. So these are the anthers. So my flower is positioned like this in the garden, on the tree. Now, if we say that pollination is the transfer of pollen grains from anthers to stigma, then now the question is, if we have anthers here with the pollen grains, these pollen grains have to be transferred from here up to here. Can these pollen grains jump from here up to here? No, they cannot jump. So if they cannot jump, then it means this flower cannot pollinate itself. It cannot pollinate itself. Why? Because the stigma is above the anthers. That's why this flower cannot pollinate itself. So the type of pollination in this flower must be cross-pollination. And what is the reason? Because the stigma is above the anthers. So that's That's the trick. Then I have another flower here. And it's appearing very tiny. I don't know if the features will be visible. Yeah, I think they will be. Now, this is also another flower here. I have it here. Now, when you look at this flower, just basic. When you look at those things in the yellow, those are now the anthers. These are now the anthers of this flower. Then what you're seeing in white is actually the stigma of this flower. No, the one in white is the filament, then the stigma is at the tip. Now still, is this one cross-pollinated or self-pollinated? I will say it is cross-pollinated. Why? Because the stigma is above the anthers. Therefore, pollen grains cannot jump and land on the stigma. But however, there are other flowers. Let me check. We'll also have another flower here. This is the morning glory flower. Let me open it and see the positioning of their anther heads and the stigma inside. So I've just opened it carefully. So this is it. Still, they may not be very visible. Now, what you see standing out, that now is the stigma. Then the anthers are now still left down. So it means that now also this one is cross-pollinated. What is the reason? The anthers are above, so the stigma is above the anthers. So that is how we are able to track and know the type of pollination a flower undergoes. Now, if the anthers are above the stigma, then that becomes the self-pollination. Because we know when the anthers produce the pollen grains will just fall down. When they fall down, they will fall on the stigma. And that is the pollination. Transfer of pollen grains from anthers to the stigma. If the anthers are above the stigma, then we are likely to get, that one is likely to be self-pollination. So that's how we reason it out. That's how we reason it out. If they ask maybe the mode of pollination, for mode, it is either insect pollinated or windy pollinated. Then if they ask you for reasons, you also give reasons. Like for example, the mode of pollination of this one, it must be insect. Why? Because it is brightly colored to attract insects, like that. Then also it produces sticky pollen grains. These yellow substances, even when I touch them with my hands, I'll be able, they will come out, they will stick. I think you're seeing something yellowish on my finger. The pollen grains will be able to stick there. So meaning now these pollen grains are sticky. So flowers which are pollinated by insects have sticky pollen grains. Whereas those ones that are pollinated by wind, they produce light pollen grains, and they are also dull colored, like that. So then another important thing on flowers, they always ask you now to describe. Maybe let us first now look at this one. They say, give 3 structural differences between specimen R and S. So they're asking for structural differences between specimen R and specimen S. Now, in practicals, when they ask for structural differences, or when they ask for different, whether in practical they will not ask you for any other differences, they always structure, because that is what you see. In practicals, they are asking you according to what you see. What's the difference between specimen R and specimen S? So for in practicals, they give you these specimens, use them, get them, hold them into your hands, and ask yourself, what are the differences, or what are the similarities? Now, of course, when you look at, for instance, these ones here, what do you notice? This one, for example, this one has 3 sepals. But before now we go maybe to the 3 sepals and what, let us first see how we describe the parts of a flower. Then we shall now be able to see how best we can write the differences. Suppose you're given these two flowers. Actually, that was the next question. So the next question is saying, examine the specimens, describe. So they want now to describe the parts of these flowers. Now, how do we describe flowers? How do we describe flowers? Now, how we describe flowers, we describe them like this. A flower has 4 distinct parts. It has corolla, and it also has the calyx. It has the stamen, and it also has the pistil. Those are the 4 main parts. We actually call them floral parts of a flower. Now, those 4 floral parts, if they ask you to describe them, this is what we do. I will start with the corolla. If they ask you to describe the corolla, the first thing you should ask yourself is, what is a corolla? What is it? Corolla is a collection of petals. So it means that now they want to describe the petals. So if they want to describe the petals, now you look at the petals and you give us their features. And the first thing is, the petals can either be free or they can be fused. That is the first thing. Are they free or fused? When you look at this flower here, I don't think that these petals are fused. You can see these petals can be separated. They are very free. So these are free petals. This is an example of free petals. You say can even easily remove one. Can easily remove one like this. So it means that now these petals are free. Or petals can be fused. Now, when you look at this flower here, of course, it has already folded. Now, when you look at this flower here, you notice that this, of course, what you're seeing are the petals. But okay, so when you look at these petals are fused. Why? They are not free like these ones here. They are not free like these ones here. Let me remove. So these ones are free. When you look at this one, they are fused. If you try to open it, still you see that these petals are fused together. They are not free. So this is an example of fused petals. I also have another flower here. It has dried and it is, but I hope now you know the difference between fused petals and free petals. Now, that is the first thing. You tell us, are these petals free or fused? Then the second step is you count how many petals are they. You count them. You count them. So for example, this one, remember also I moved this one from this flower. So this is petal number 1. I want to remove as a count. This is petal number 2, petal number 3, petal number 4, petal number 5. So it means that this flower has 5 petals. So then also you comment on the color. Now, the color of flowers, we don't give the actual color as we see it, whether red, pink, or yellow, or blue. Flowers exhibit 2 colors. They are either brightly colored or dull colored. So these petals are brightly colored. So those are the 3 ways how you can describe petals. So what have I said? that this flower that I had, this flower here that I had, this flower, I'm describing its corolla. I said that the first thing is to know the corolla is made up of what? It's made up of petals. It means that now I have to describe the petals. So I will say this flower has 5 petals, has 3 petals, has brightly colored petals. So I have described the corolla of this flower. Then what of this one here? This one here, however much the petals are fused, you can still count and see their number. Like for example, now this one, you have to count how many petals are these. However much they appear to be fused, they can easily be counted. They can easily be counted. These ones here, you can count them. And if you count them, let me try to count. It is closely closing. You know, there are some flowers which close during the day and open at night. No wonder that's why this flower is called the morning glory. And of course, in coordination in plants, you know this type of response, the one we call the nastic response. Now this one is photonastic. Maybe it fears light. The theory, they can ask those things in the theory. Okay, so now this, when you count these petals, they are 5 in a number. You count, you don't have to cram. For you, any flower that they give you, use that specimen and study it carefully and ask the questions that are being asked. So this one, the petals are 5. They are fused. The color, I don't know if purple is brightly colored or dull colored. I don't know. But to me, still, this color is bright enough to attract bees. So this is still a brightly colored flower. So it means that now I will say, now I want now to describe this flower here. I will say has 5 petals, has fused petals, has brightly colored petals. That is how I describe the corolla, these flowers. So look at the question. The question was saying, describe the corolla of specimen R. I have described the corolla, haven't I? I have. What did I use? I asked myself, what is the corolla made up of? The petals. So it means that now I have to describe the petals. Then if they ask you now to describe the calyx, then you have to ask yourself, what is the calyx made up of? The calyx is made up of the sepals. The sepals are the green parts of the flower. Those green lower parts are what they call the sepals. That green. Now, it means that now here you have to describe the sepals when you're describing now the calyx. Now, how do I describe these sepals? Still, I can count them, how many they are in a number. After counting how many they are in a number, then are they free or fused? You'll still be able to see whether they are free or fused. You can see them. Then also you comment on their color. Now, in flowers, green is a dull color. So how am I going to describe the calyx of this flower? I will say it has 5 fused dull colored sepals. So I have described this, the calyx of this flower. So that is how we describe those things. So then, what of now the pistil? Because some questions can say, describe the stamen, describe the pistil. Still, the trick is in knowing these parts that they want us to describe, what are they made up of? What are they made up of? Then if you know what they are made up of, then it means you'll now have to describe the parts that make up those, that make up the section that you're talking about. For instance, when you look at our question here, look at Roman number 2. Roman number 2 is saying, describe the androecium. The androecium. So you have to ask yourself, what is androecium? and what is it made up of? So if you ask these questions, then you'll be able now to describe. Now, for instance, the androecium, the first thing to ask yourself, what is the androecium? Androecium, these are the male parts of a flower. The male parts, that is the androecium. If it is not called androecium, it can be called stamen. So another question can come, describe the stamen. So ask yourself, what is this stamen made up of? So the stamen is just the anther and the filament. That is the stamen. So it means that now you are now describing the filaments and you're also describing the anthers. So for instance, when I look at this question here, those very many thread-like things you see with the yellow heads are actually the filaments or the stamen or the androecium. Now here I have to describe this. Now, how do you describe it? You also count how many are they. Are the filaments thin? Are they thick? Are they short? Are they long? Are they brightly colored? Are they dull colored? All those things you ask yourself. Then you go to the anther head. Is the anther head brightly colored? Is it lobed or not lobed? Like that. Now, for instance, when you look at this flower here, I now want to describe the androecium of this flower. Of course, this is the hibiscus. It is a very common flower. So I want now to describe the androecium of this flower. I've said that now because I know that the androecium is made up of the filaments and the anther head. So it means that now I'm describing the filaments and the anther head. So I will say it has many filaments. The many is coming from where? Counting the number. If you look at these filaments, there are too many to be counted. So if there are too many to be counted, you just report it as that it has many filaments. When you look at this flower, this one here, the morning glory, you open it to expose the stamen. You open it to expose the stamen. You can remove the petals carefully. Then now you look at now the filaments that you want to describe. So you count them. These ones are few to be counted. They can be counted. So you count them. How many are they? For instance, this one, I'm seeing 5 filaments. So then now when I'm describing the filaments of this flower, I will say has 5 filaments. Then, are these filaments thin or thick? There are some plants which have, there are some flowers which have very thick filaments. So I will say has thin filaments. That is also another description. Then you look at now the size. Are they long? Are they short? When you look at the filaments of the hibiscus flower, for example, these ones, these filaments are very short. They are very short. So we'll say has short filaments. But when you look at this one, these filaments, they are of varying sizes. There are those ones which are long, there are those ones which are short. So you can say has filaments of varying length, like that. What you see is what you tell us on paper. Then the anther heads. When you look at this flower here, then you see that the anther heads, they take up a very funny shape, like boat-shaped, like arrow. But when you look at them critically, you see that these anther heads are long and they are lobed. So I'll say has lobed anther heads, like that. If they are showing some distinct color, then you also say the anther heads are brightly colored. Like for example, this flower here. When you look at this flower, like I told you, those things that you see appearing yellow, those are actually the anther heads. So we shall say has brightly colored anthers. Now, you can call them anthers or you can call them anther heads, whichever way you are correct. that way. Then also now for example, a flower like this one here, you want to describe its androecium. You look at those anther heads, all the anthers, you count them, how many they are. For example, now this one, I'm seeing 5. Then now you look at the filament. How is their filament? Now, when you look at a flower like this one here, actually what you're seeing in yellow, these are now the anther heads. The filaments may not even be visible. But when you try hard to open it, you see that it has very short filaments. So it means that now when I'm describing this flower, I'll say it has short filaments. Like that, has short filaments. That is how we describe the androecium or the stamen. Then I can ask you now to describe the gynoecium. Another name for the gynoecium is the pistil. So still, you have to know what are the parts of the pistil. So those are the parts that you have to describe. The pistil is made up of the stigma, the style, the ovary. Those are the parts of the pistil that you can see from outside. So it means that now you're going to describe those parts, those 3 parts. Describe the stigma. How is it? Count. There are some flowers which have a single stigma. You say it. There are some flowers which have a bilobed stigma. There are some flowers which have, bilobed means 2 lobes. There are some flowers which have many lobes. For example, look at this one here. Now, the red heads you're seeing, those red heads, that is the stigma. So it means that now this flower has how many stigma? They are 5 in number. So when I'm describing now the pistil of this flower, I'll say has 5 stigma, like that. Then you also comment on the filament, sorry, the style. For example, this flower, now the style of this flower is actually inside. You have to open. Let me open it and I show you. So you peel off that stamino tube carefully. Then you pull out. So this, what I'm pulling out is actually now the style. So you see how my flower is? So this one is the stigma, the red. So the stigma is brightly colored. Then it has now one style. See this thin thread-like thing. Now these are the stigma. This is the stigma. So it means that when I'm describing this stigma, I will say has a long style, has a thin style. So that is how I will describe my pistil of this flower. Has long style, has a thin style, like that. So that is how we describe the parts of a flower. Just what you need to know is, do you know the parts that make up that particular part that they're asking you to describe? Okay, and then part E is saying, give the advantage of the arrangement of the essential parts of the specimen over that of R. Now, here they're saying the essential parts. Essential parts. Now, what are essential parts? Now, a flower, its role is for reproduction, is to reproduce. That's its role, to reproduce. Now, if they say the essential, then it means that now, when they say essential parts, you remove all those parts that you think are not essential. Then you remain with the parts which are essential. Now, the petals on a flower, we consider them as non-essential parts. So you remove them. So I'm removing the non-essential parts of a flower, like this one. I remove the petals. Also, my calyx is still remaining. The calyx is also non-essential. I also remove the calyx. So what I have now remained with is a flower showing only the essential parts. Now, they can ask you to draw this. You sketch, you draw it, you draw it. So, but now they're asking, let me go back to the question, see what they're asking about. They're saying, give the advantage of the arrangement of the essential parts of specimen S over that specimen R. Now here, we may not be able to tell, I don't want to tell you that maybe specimen S is the morning glory, or specimen R is the hibiscus flower. But now, what does that question ask? What is that question asking for? They're asking for, when you look at maybe these two flowers, when you look at them, the arrangement of this, or the arrangement of these parts, how is it of an advantage compared to the arrangement of the parts of this flower? Like that. For instance, you'll see if maybe here the anthers are brightly colored, and the other one, the anthers are not brightly colored, then what is the importance of the bright color of the anthers? Like that. Then there are some flowers like this one. Find now the essential parts are enclosed inside the petal. But when you look at a flower like the morning glory, so the hibiscus, this one that I'm from dismantling, the parts were exposed outside. Now, what is the importance of that? This one, where the parts were exposed outside, the plant can, the filaments, so the stigma is exposed to the external conditions or the external environment for easy pollination. So it is easy for pollen grains to be carried by insects from this to another flower. So look at this one, where they enclose their things inside, it makes it hard for the pollen grains to be transferred one flower to another. So basically that is what you look at. You look at the special feature the other flower has that is not in this one, then now you give us the importance of that special feature that you have identified. So it means that now that will be the advantage that this flower has over the other, like that. Then of course, these questions will always ask you something about drawing. So still, we may not go into the details, maybe the actual drawing of this one here. But what do you need to know when they ask you to draw things? When they ask you to draw things, first of all, in practicals, you must use a pencil. And the pencil must be a sharp one. The pencil used must be a sharp one. Then, when you're making your drawings, one, start with the title, write the title. And the title should be a drawing of, or a drawing showing, this and this, having all the instructions. For instance, let me give you this and we see the possible title to this one here. Now, in this one here, the possible title, because remember I say that remove all the sepals and petals of specimen R. Draw and label the remaining part. Set your magnification. Now, what would be the title should actually be telling me whatever I have done. That's what the title should be able to tell me, whatever I have done. So if I say that a drawing of specimen R, that title is wrong. Why is it wrong? Because that is not how R looked like. Because remember they said remove all the sepals and the petals, draw and label. So now my title should actually be reflecting what I have drawn. So in this case, maybe you can say a drawing showing specimen R without sepals and petals. Or you can say a drawing of specimen R with the sepals and petals removed. In other words, if there are special instructions in the question about how you should prepare your specimen before drawing it, then those instructions should also be reflected in your title. So you have to be aware of that. Then, another thing about the drawing is the magnification. After making your drawing, then you must do the magnification. For instance, let me project another item. You see what I'm trying to mean here. Do I have it here? No. I think I forgot to put it on the computer. I think it is this one, yeah? Now you look at my drawing here. Look at my drawing. Now, look at the question. The question is saying, remove all the sepals and petals. And look at how my title is, how my heading is. Drawing of the remaining part of specimen R after removing all the sepals and the petals, like that. So you find that in my heading, even some part of the instructions is there. If maybe they had said that remove the non-essential parts, draw and label the remaining part, then it means that now even your heading would be a drawing of the essential parts of specimen R, or a drawing showing the essential parts of specimen R. In other words, your heading should always be reflecting some bit of the instructions that you followed when you're preparing your specimen for the drawing. There are some questions that, for example, they can, if maybe it is a cockroach. I've ever seen a question on a cockroach. They're asking the learners to cut off the leg, the hind leg. After cutting off the hind leg, then they say, draw and label the first 3 segments of the hind leg. The first 3 segments of the hind leg. So what does it mean? It means that now you have to only draw the first 3 segments of the hind leg. You don't draw the whole leg. And also in your title, that statement must be reflected, that maybe a drawing of the first 3 segments of the hind leg of specimen R. So like that. That is how your title should always be. If there are special instructions, also those special instructions should be embedded in your heading or in your title. Don't use the word a diagram of. It is not a diagram. It is not a picture. Just say a drawing. Can say a drawing showing the remaining part, or a drawing of the remaining part. Like for example here, then your drawing should have the magnification at the bottom right-hand side. Your drawing, you must put there the magnification. Magnification, you just write there one word, either times 1, full stop, or times 2, or times 5, just that. You don't put there that magnification is, no, just put there times the figure and you stop there. Now, how do we determine the magnification? Do we need the ruler? We need a calculator to get the magnification? No, you don't need a calculator, you don't need a ruler. What you have drawn on your piece of paper, get your flower, get what you have drawn, make it sit there on your paper, and you compare how big is it, or how big is your drawing compared to the actual specimen, or how big is your specimen compared to the actual drawing. You compare. Then if you see it is 2 times, then means that the magnification will be times 2. If it is almost the same size, then the magnification will be times 1, like that. So magnification, you can just estimate it using your eyes, or you can put the actual image or the actual specimen on the paper and you see how big your drawing is. Then because for us the magnification tells us how big or how small your drawing is compared to the specimen that we gave you. If I ask you maybe, how big is your house at home compared to the class that you are sitting in? If I come and tell you that maybe my house at home is 3.78 times bigger, you'll get confused. You say, now, why is he using the 7.8, 3.788 what? Let them be whole numbers. Let them be whole numbers. Maybe it is 2 times 2. Unless if maybe it is, maybe what you have done is too small compared to the actual drawing. Then now the magnification can now go to things like times 1.5, or maybe times 1/2, like that, or times 0.5, like that. So your drawing should always be having the magnification. Then the labeling should be clear. The label lines should touch the actual part that you are naming. For example, when you look at my drawing here, the label lines are actually touching the actual part. They are touching. They are not stopping along the way. They are touching. So if they are touching, that is the correct form of labeling. Correct form of labeling. Then yeah, I think that is what we have to know about drawings. Then I think now we are almost done with everything about the practicals. And remember now, for number 2, if it is a paper, for number 2 is always, sorry, number 3 is always still the same like number 2. It is about anatomy still, the structures. Number 2 was plants. Now number 3 can be on animals. They bring you any animal part, or they can bring you any animal. Maybe it can be an insect. It can be any arthropod. It can be a millipede. It can be an earthworm. Anything. Then so they ask you things of observation. Observe, describe, draw those things. So we may not go into such details, but I believe that the skills I have given you in answering number 2 can now transfer those skills answering questions also of number 3. When they say observe the features, for example, in this one, if maybe V was let me say maybe a honeybee, or maybe if V was a termite, or maybe if V was a housefly, maybe a cockroach. Let me use a cockroach because a cockroach is always bigger. If V was a cockroach, and they're saying, basing on the, maybe they're saying, describe the features of on the head describe the features on the head of the cockroach. Then now I look at the cockroach, I look at the head. Then now I look at the parts of the head. Then I now start describing those parts of the head. I keep on now describing those parts of, I now describe those parts of the head. For example, if I look at a cockroach, I know that a cockroach has antenna. Then it means that now I have now to describe the antenna. I don't just have to say that the specimen has antenna. You've not described the feature. How do the antenna look like? Has long antenna, has segmented antenna. The antenna is tapering towards the end, like that. So that is how we describe the features. We must attach something that I should be able to read and be able to understand what actually you're looking at. If it is the compound eyes, don't just say that the specimen has compound eyes. What about those compound eyes? What about them? Maybe the specimen has large compound eyes. Now, using the word large is the descriptive word that we are looking for. Using the word long, that is what we are looking for. Segmented is what we are looking for. So then, if maybe they say now, maybe describe the features of the legs. So maybe describe the features of the thorax. Now, I have now to look at the thorax and I ask myself, what is there on the thorax? What is there on the thorax? What is now? It means that now I'm going like for flowers. I told that for flowers, if they say describe the corolla, then you ask yourself, what is the corolla? What is it made up of? It is made up of petals. Then it now means I have to describe the petals. Now, if they say describe the features of the thorax, now it means that I now have to look at what is on the thorax and I describe those parts found on the thorax. For instance, if it is a cockroach, I know that the thorax, first of all, the thorax has legs. The thorax has wings. So it means that now in this question, I have now to describe the legs. I have to describe the wings. How do they look like? That's what it means. If they say describe the features of the thorax, the question means that how do the features of the thorax look like? If you say it has legs, how do the legs look like? It has wings. You've not described how do the wings look like. So you tell us that it has 3 pairs of legs. Now, the 3 is the description. You go on and tell us that maybe the legs have sharp spines. The sharpness is the description. The legs are jointed, or the legs are segmented. Now, the word segment or the word jointed is the description that we are looking for. The wings, what is so special about the wings? Has maybe a pair of outer wings. A pair is the description. The outer wings are hard. Using the word hard is what we are looking for. Hard is the description. The inner wings are broad, a description. The membranous is the description. So that is how you describe. The word describe means how does it look like? Like that. Then they can now ask you now to give adaptations. Maybe they can ask you, how are the legs of the cockroach, for instance, adapted to their mode of life? Or how are the legs of a housefly adapted to the mode of life? So it means that now you have to look at now, when you look at the legs of the cockroach, how are they adapted? Or what special feature do they have? The legs of the cockroach, first of all, we said they have sharp spines. Now, we want now to know the function of the sharp spines. It has sharp spines for defense, like that. The legs are segmented. What is the importance of the segments? It is segmented for flexibility during walking, like that. Maybe the ones of the housefly, the legs have hairs. How is it adapted? The legs have hairs to carry germs, like that. So that's what they mean when they say describe, and also maybe they say give adaptations. Now, adaptations for every part that you state, still that part must be having a descriptive term on it. Then you give the function. If possible, the function should be much of the descriptive term that you have used. For instance, if you're giving us adaptations of the compound eyes, and you said that compound eye has large compound eyes. Now, we shall ask you, what is the importance of the large compound eyes? And we are emphasizing the word large, because it means that you have seen that what is so special in these compound eyes is the size of the compound eyes. Then you should be able to say it has large compound eyes, which increase the field of view. So increase the field of view. Don't just say has compound eyes for seeing. Any eye can see. But what is so special with these compound eyes on that insect? They are large. Then what is so important about them being large? To see a wide field of, to see a wide area, like that. Has long antenna. Now, for what? Now, the function should be related to the length, that has long antenna to sense over a wide area, like that. Has segmented antenna. What is the importance of the segments now? Has segmented antenna for flexibility, or to easily move the antenna to all directions, like that. So when you're giving these adaptations, just know that the function you're giving should be attached to the descriptive word that you have used. So that's how we give adaptations, like that. Then what else have I not talked about? I think I've talked about most of the things. Now, we're remaining with only 5 minutes to end our session. I think we may have to extend it for about 10 minutes, maybe, depending on if my battery can hold that long. Because it seems the battery had charged it to time the 2.5 hours of our discussion, and now it is warning. Now, I'm going to entertain questions. Now, the questions are now open. Any area in the practical paper. Still, you can keep them flowing in the chat. I've already identified some questions there. Let me go there and see if I have questions there. Okay. Nalwayo SS, they are asking, is it correct to say anther head, or one simply says anther? I believe there is no anther type. Now, anything you want to use here is fine. You can either say the anther or the anther head. What are the things that I must put under consideration when drawing a diagram of a flower? Now, There are no special things to put under consideration when you're making the drawing of a flower. But maybe the question would be, what are the things you should put into consideration when making a biological drawing in a practical paper? And they have given you most of those tips. I think this question was asked before. I had given you how to draw. So all biological drawings in a practical, use a sharp pencil. The title must be there, and I've given you the tricks how you can answer, how you can rate the title. If there are special instructions, those special instructions must be reflected in your title. Then your drawing must be having magnification. I've given you a small hint about how magnification can be written. Then the labels, the label lines must touch the exact part that you are naming. Then also we are gathering feedback to see the effectiveness of these programs. So if we have teachers together with their learners, you can kindly ask the learners what they liked or disliked, and the way forward for the next set of learners. You can maybe still put your comment in the chat, or you'll forward that comment on the WhatsApp platform where you picked today's link from. So feedback is very crucial to us. So please, you can rate the feedback, what you have liked, or what you have disliked, that it can give us guidance on the next engagements. Then there is a question that if it is the instruction in the question on magnification, I've not now got this question. Different vocation and secondary school. If it is instruction in the question on magnification, I've not got the question very well, but I hope you have now known magnification, how it is got, and where to write it. Springfield, they're asking, what could be the reason for identifying the phylum of an organism as chordata? Well, now, chordata, you ask yourself, what are the chordates? Now, the best way to understand where these reasons are coming from, you think of an example. What is a chordate? We are chordates. I'm a chordate myself. And then a goat is also a chordate. Now, which features do I share in common with a goat? Those features that I share in common with a goat, now those are the reasons why we are all put under phylum chordata. That is the trick. When you look at yourself, when you look at yourself and you compare yourself with a goat, is that now all of you have a backbone. So if all of you have a backbone, then that means that is the reason why you're all put under chordates. Backbone, notochord, that. Then in two pairs, limbs, the things that you share in common with another organism, those are the things that put organisms under a given category. Though I've not seen questions related to this in practical. Then, when labeling a drawing, are we to use curve or brackets? Curve or brackets. Now, when you're labeling, please don't use brackets. The way you saw how I labeled the other drawing. Now, sometimes things are practical. We always emphasize not to use a lot of creativity. We always emphasize it. Now, the way you see, apart from maybe only that, apart from drawing using a pen, the others do as they have done here. If you have any other alternative, please don't do it. Don't use brackets anywhere. Don't use arrows anywhere. Like that. Then, so Kasiasa Secondary School, I hope I have answered you. Okay, then would it be okay conclude that hydrogen peroxide is broken in case where it is provided as an unknown, let's say Q. Now, if hydrogen peroxide is given as Q, then for you, you maintain the identity of whatever you have been given as Q. So you can say Q was moderately broken down. Q was rapidly broken down. Or you can say there was rapid breakdown of solution Q. It is better to maintain the letters given to the solutions. Unless they have told you that the solution Q is hydrogen peroxide, then now you can now say hydrogen peroxide is broken down rapidly, slowly, or moderately. So maintain the identity. Then, may you please talk about the dichotomous keys? Now, the dichotomous keys, those ones, is also another big area. And it's like we would use about still 30 minutes to talk about them. So those ones, I'm very, very sorry. Maybe if I had prepared for this, if you had some grading question, I'd have done it. But now, kindly, that the school is not reflected, Chrysalis, I will now refer you back to your teachers. You can go to them and they guide you more on the dichotomous keys. We cannot have enough time today to go through them. Then Samuel Cornerstone, I don't have what's expressed. Okay, thank you. Thank you for the feedback. Maybe also say something about fish. I've seen it in one of this year's mock exams. Now, still fish can now come as a substitute in number 3, where they're asking about where they're setting about animals. Now, fish, still it takes us back to the theory. Fish can come from the component of gaseous exchange. Fish can come from locomotion. So still on the fish, they can now ask, maybe pull out the gills. How are they adapted for gaseous exchange? That is the theory. They are moist. They have very many filaments. Then you draw. They can maybe ask you to draw them. Then maybe they can now ask you to comment, to look at the scales. Touch the scales. You find that they are hard. Then they ask you, of which advantage is that to the specimen? They are hard for protection. They offer protection to the organism or to the fish well in water. They are slippery to reduce resistance when swimming, things like that. Then, like if they said draw the diagram in specific magnification, like ×5. Now, please, they cannot ask you to draw a diagram and then they dictate the magnification. That one they cannot ask. They cannot ask a diagram, then they dictate the magnification. Now, is number 2 only about flowers? Now, please, number 2 is not about flowers. I picked a paper to guide us in our discussions. And by coincidence, in this paper, my number 2 happened to be about flowers. But number 2 is about plants, things from plants. That is number 2. We call it botany. Botany. And in this botany, they can ask you about flowers. They can ask you about leaves. They can ask you about fruits. They can ask you about storage organs. They can ask you about stems. And all of this, apart from maybe the flowers, all these are mainly coming from one topic, structure of flowering plants. And that is a senior 1 topic. So these are the things that can come on number 2. It can be any of these: flowers, leaves, fruits, storage organs, and stems. The number 3 is not only about insects. Number 3 can be birds, it can be fish, it can be arthropods. Under arthropods, we have the insects. It can be teeth, it can be bones, all those things. It can be about any of those. So it is not, sorry, I forgot to share the screen. Okay, so this is what I was talking about. Number 2, that it can be about any of these: flowers, leaves, fruits, storage organs, and stems. Can be about those. Then the number 3, it can be about fish. It can be about feathers. It can be about bones. But feathers, now we are following the abridged. Feathers were abridged, so you don't expect them to set from the feathers unless otherwise, because feathers were abridged, and also which other word? Also the bones were also abridged. These bones here, the vertebral column, those ones were also in the abridged. So you not expect to find a practical, or even if in theory, in a paper, in a question to deal with feathers, or locomotion in birds, locomotion in fish, locomotion in insects, and also these vertebral bones. Those ones were off. They were removed off the abridged. So you should focus on other things other than those ones. Okay. Just a moment, it tend to have lost connectivity. The phone I was using has switched off. So let me first connect, then I conclude. Let me pass. Okay, sorry for that brief. I hope you can now hear me clearly still. Okay, so I was concluding that those are the things that you should expect. I've briefed you about what was abridged. Those ones. Okay, let me see if there are other questions to that team. Okay, is it a must to label drawings on one side only? No, it is not a must. You can label from any of the sides. So there are no rules to that. Basing on your observations, state the class to which specimen S belongs. Give 2 reasons to support your answer. If labeling are many on a diagram, which direction should the labeling be? You just make sure that you draw an organized drawing. You find where you find an appropriate direction or position to put put your labels. There are no rules that they must be in a specific place. As long as at the end of the day, the drawing is your drawing looks neat. Remember, there is always that mark for neatness. So I think we can stop from here. We can end from here.
Teacher Arthur.
Yes, Mr. Kazim.
Yes, there was a burning question. I don't know whether Wampeo, I think it was Wampeo. Wampeo, you had a question. I don't know whether they are still on. Wampeo, Take, you had a question. Wampeo, there was a question you wanted to ask. Wampeo, is it possible to unmute Teacher Henry? I think I'm failing to get to Wampeo. They had some question they wanted to ask. Okay. Okay. Okay, I think we are failed to get Wampeo. I don't know whether there is any school. Teacher Arthur, is it possible to throw some little light on the other paper one? Just light and maybe some guidance to the contents.
Well, paper one is basically the theory. And you just have to read widely in all areas. The things, the nitty-gritties of how to answer them, they're also wide. But those are some of the things, they're the ones we've been engaging in our previous sessions. So the only thing I can say that now the learners, they should now sit down, read or revise strategically in this remaining 1 week, targeting the areas where they feel that they are still, and also making use of your teachers. And also try to look at the questions in the different mock exams. Look at the questions in different mock exams and see if you can attempt those questions in this remaining 1 week. Yeah, briefly, that's what I can say for now in relation to the theory. You must be having now the content. You must be having the content. And time is always enough. You can prepare adequately this remaining week to do something helpful. Okay, so Mr. Kazivandi, Mr. Yes. Dungo, I think we have come to the conclusion. I think if you have any closing remarks, you can give them. Otherwise, I feel I have tried to cover most of the relevant areas. Thank you.
Okay, thank you so much, Teacher Arthur, for the wonderful presentation. We are really very grateful. Thank you so much. I want also to thank the schools that have been able to join this call. Thank you so much. Kasese, okay, Teacher Arthur, maybe there is that number 31. I feel there is something you may need to add on. Kasese Mozim is asking about that number 31 in paper 1.
Mr. Kazut, now, that number 31, actually, most of our previous sessions is the number that we've been focusing on. It is a major area, and that one needs almost about 3 hours alone. So I cannot even mention anything about, even if I'm given 10 minutes to comment about it, those 30 minutes cannot even be sufficient enough.
Okay, Teacher Arthur. But I feel maybe Teacher Arthur, what we can do, we can share with them the questions from Abby.
Yeah. That one, I'm going to forward you the the questions from the different seminar engagements we have had all over the country. And we always actually focus so much on that number 31. So I think it will be of help. I'm going to share those things with you. Then you'll forward them to the groups where they picked the invite from.
Okay. Okay, I think we are going to do that. It will really be of great help. Okay, thank you so much once again, Teacher Arthur. We want to thank all the schools that have joined in from the different parts of the country. I'm going to allow schools to first put on their videos. Those schools that are having their students, kindly have your video on. I want us to take a photo before we go into the last part of this session. Kindly just have your video on. All those schools that are having students, have your camera on. I want us to take a photo. Okay. Just keep your video on. We are going to take a photo. Okay. Teacher John Ngatu, I'm going to ask you to unmute and just say something small before we come to the closing of this session. Yes, Mr. Ngatu, just prepare something small for us. At least even if it's a greeting, it will be enough. So I want to take some photos for the schools that are online. Okay. Okay. Thank you so much, all those schools. So I'm still taking some photos. Thank you so much. Thank you so much, all those schools. Kasese Secondary, Pakada, Moyo SS, Star SS, Makerere College, Popo Anak, Zeo SS, Sir Samuel Baker, and I'm going to ask the teachers to use your phones to take some photos of your students when they are attending the session. But before we close, I want to invite, there is a teacher I want. What is, oh, has it gone? Oh, I wanted them to say something. There's a teacher had asked. I don't know that is still on. Teacher John, are you still on? I wanted you to say something. I think I've lost him. But thank you so much, teachers. Take some photos of your students who have attended the session, such that we can be able to have them in the report that we are going to submit to the Ministry of Education and make more recommendations. But now allow me to thank all the schools that have joined, all the teachers and all the ED teachers that have been able to connect their students to this session. Thank you so much. Allow me now to invite the principal at the Ministry of Education and Sports, Mr. Ronald Dungu, to have his closing remarks and finally close this session. Mr. Ronald, you are most welcome. Mr. Ronald? Mr. Ronald, something, I don't know whether you have some technical challenge. Oh, okay. I have unmuted you, Mr. Ronald.
Yes, Mr. Kaziba, thank you for running us through today. I want to thank Teacher Arthur for ably hosting us as schools and teachers. I have enjoyed everything. I have relearned my biology and how I wish I was a student. So I also hope that the students have learned a lot and they are now going to prepare well for the final exams. We want to thank the schools, the teachers who have made this possible for the students to be here. To the candidates, we want you to be the very best because you are the last we are having in this curriculum. We really, really want you to do your best, perform at your best, so that we wind up the old curriculum with very good remarks. With that, I want to wish all the candidates success in your exams. And to the teachers, thank you for running the show up to the end of the year, because now you are handing over the students to UNEB, and we will all wait to celebrate the results that will come. I want to thank all of you for joining, and to that, I want to declare this session closed. Bye-bye.
Bye-bye, everyone.
Bye-bye.
Kasese Secondary, let me hope you have taken a photo before those students leave. Dear teacher, take a photo of Kasese Secondary School. Before those students leave, take a photo and share with us such that we can document. It's really so nice to have Kasese Muslim Secondary School. Moyo SS, thank you so much. Makerere College, thank you so much. Namilyango College, thank you. Pajule and Samuel Baker, thank you so much. Okay. Thank you so much, dear teachers. We shall share the recording, and bye-bye.